Let's talk about conformational stability and how this relates to protein folding and denaturation. So first, let's review a couple of terms just to make sure we're all on the same page. And first, we'll start out with the term conformation. And the term conformation just refers to a protein's folded 3D structure, or in other words, the active form of a protein. And next we can review what the term denatured means when you're talking about proteins. And denatured proteins just refer to proteins that have become unfolded or inactive. So all conformational stability is really talking about are the various forces that help to keep a protein folded in the right way. And these various forces are the 4 different levels of protein structure. And we can review those briefly right here. So recall that the primary structure of a protein just refers to the actual sequence of amino acids in that protein. And this is determined by a protein's peptide bonds. And then next you have secondary structure, which just refers to the local substructures in a protein. And they are determined by backbone interactions held together by hydrogen bonds. Then you have tertiary structure, which just talks about the overall 3D structure of a single protein molecule. And this is described by distant interactions between groups within a single protein. And these interactions are stabilized by van der Waals interactions, hydrophobic packing, and disulfide bonding, in addition to the same hydrogen bonding that helps to determine secondary structure. And then quaternary structure just describes the different interactions between individual protein subunits. So you have the folded up proteins that then come together to assemble the completed overall protein. And the interaction of these different protein subunits are stabilized by the same kinds of bonds that help to determine tertiary structure. So all of these levels of protein structure help to stabilize the folded up active conformation of a protein. So why is it so important to know about the different levels of protein structure and how they contribute to conformational stability? Well, like I said, a protein is only functional when they are in their proper conformation, in their proper 3D form. And an improperly folded or degraded, denatured protein is inactive. So in addition to the 4 levels of protein structure that I just reviewed, there is also another force that helps to stabilize a protein's conformation, and that force is called the solvation shell. Now, the solvation shell is just a fancy way of describing the layer of solvent that is surrounding a protein. So say I have a protein who has all these exterior residues that are overall positively charged. And picture this protein in the watery environment of the interior of one of our cells. Then the solvation shell is going to be the layer of water right next to this protein molecule. And remember that water is a polar molecule, so you have the electronegative oxygen atom with a predominantly negative charge, leaving a positive charge over next to the hydrogen atoms. The same is true for each of these water molecules. So now, as you can see, the electronegative oxygen atoms are stabilizing all of the positively charged amino acid residues on the exterior of this protein. So as you can see, the conformational stability of a protein depends not only on all of these interactions that contribute to primary, secondary, tertiary, and quaternary structure, but also what sort of environment that protein is in. And all of these interactions are very crucial for keeping a protein folded properly so that it can do its job. Now, what happens when things go wrong? How does a protein become unfolded and thus inactive? Well, remember that this is called denaturation, and this can be done by changing a lot of different parameters within a protein's environment, including changing the temperature, the pH, adding chemical denaturants, or even adding enzymes. So let's start with what happens if you alter the temperature around a protein. And we can use the example of an egg when we put it into a pot of boiling water. Because an egg, especially the white part, is full of protein. And this pot of boiling water is representing heat. And remember that heat is really just a form of energy. So when you heat an egg, the proteins gain energy and literally shake apart the bonds between the parts of the amino acid chains. And this causes the proteins to unfold. So increased temperature destroys the secondary, tertiary, and quaternary structure of a protein. But the primary structure is still preserved. So the takeaway point is that when you change the temperature of a protein by heating it up, you destroy all of the different levels of protein structure except for the primary structure. So now let's say you were to take an egg and then add vinegar, which is really just an acid. The acid in the vinegar will break all the ionic bonds that contribute to tertiary and quaternary structure. So the takeaway point when you change the pH surrounding a protein is that you have disruption of ionic bonds. And if we think about this a little bit more deeply, it kind of makes sense because ionic bonds are dependent upon the interaction of positive and negative charges. So when you add either an acid or a base, which in the case of an acid is just like adding a bunch of positive charges, you kind of disrupt the balance between all of these interactions between the positive and negative charges within the protein. So now let's look at how chemicals denature proteins. Chemical denaturants often disrupt the hydrogen bonding within a protein. And remember that hydrogen bonds contribute to secondary, tertiary, all the way up to quaternary structure. So all of these levels of protein structure will be disrupted if you add a chemical denaturant. So let's take our same example of a protein with an egg. And say if you were 21 years or older, you got your hands on some alcohol and you added this to the egg. Then all of the hydrogen bonds will be broken up, leaving you with just linear polypeptide chains. And then finally, let's take our hard-boiled egg from the temperature example, and let's eat it. So here's my beautiful drawing of a person representing you eating this hard-boiled egg. Once the egg enters our digestive tract, we have enzymes that break down the already denatured proteins in the egg even further. They take the linear polypeptide chain, whose primary structure is still intact, and they break the bonds between the individual amino acids, the peptide bonds, so that we can absorb these amino acids from our intestines into our bloodstream, and then we can use them as building blocks for our own protein synthesis. And that's how enzymes can alter a protein's primary structure, and thus the protein's overall conformational stability. So what did we learn? Well, we learned that the conformational stability refers to all the forces that keep a protein properly folded in its active form. And this includes all of the different levels of protein structure as well as the solvation shell. And we also learned that a protein can be denatured into its inactive form by changing a variety of factors in its environment, including changing the temperature, the pH, adding chemicals or enzymes.